Hi, my name is Isaku Tanida with Element Biosciences. Today we're going to walk through a detailed study performed with our partners at Olink. We successfully combined the use of their proteogenomics platform, Olink Explore, with our AVITI system as NGS readout for their assay. To begin, let's do a quick review of the Explore platform and its basis in PEA technology. PEA stands for Proximity Extension Assay. It uses the binding of two unique oligo-labeled antibodies in proximity to one another to identify the bound marker. In Olink Explore, the resulting double-stranded DNA bridge that's formed is PCR amplified and sequenced by NGS as its readout. This enables parallel analysis of multiple protein biomarkers at exceptional specificity and sensitivity with minimal input material. Currently, Olink Explore works successfully on several NGS platforms, so the goal of our collaboration was to simply further expand these options by determining its compatibility with the AVITI system. New technologies like Olink Explore that use NGS as readout has further increased the need for novel sequencing solutions that deliver high data quality at low cost with user flexibility. The AVITI system provides such a solution, with performance features that make NGS more accessible to labs than ever before, with its best-in-class accuracy, exceptionally low operating cost, and its independent dual flow cell design enabling time-saving efficiencies. In order for us to determine the compatibility of the AVITI system with the Olink Explore platform, we studied key performance metrics that include correlation, precision, sequencing quality, and output. Similarly, from a usability standpoint, we also wanted to ensure that the adoption of the protocol using the combined platforms would be easy and highly reproducible. We began by generating 4 libraries from a selection of plasma samples and pools of recombinant antigen standards using 4 384-plex Olink Explore panels, you can see listed here on the slide. And a 5th library was prepared from a common pool of PCR products from a healthy human plasma sample and split into separate further PCR reactions where 96 different sample indices were incorporated. The libraries would be purified and first sequenced at Olink using the NovaSeq 6000 SP flow cell and then sequenced on the AVITI system. All libraries sequenced on the AVITI were circularized as part of the preparation process using the Element Adept Library Compatibility Kit. This workflow is shown at the top in green. The Adept protocol is simple and takes only 75 minutes to complete, requiring no PCR amplification. FASTQ files generated from all sequencing runs were processed and analyzed at Olink. So let's take a look at the results. Here you can see the AVITI data plotted versus the NovaSeq data, based on the number or counts of each protein marker identified by each respective platform for each panel. You can see by the tightly clustered line that the number of match counts were comparable between the two platforms, the correlation being very high, at a measure between 0.984 and 0.989. For the lowest counts in the lower left corner of each of the plots, you can see some increased variation, but that's not unusual and to be expected. It's important to add that this analysis was done prior to any additional normalization treatments of the data. So in the next slide, you'll see the same data plotted after normalization has been applied, where we expect the correlation values to increase. And here you can see the expected improvement in correlation after normalization, with the data points cluster along a narrower line and scores rise to 0.992 to 0.996. Normalization is an Olink standard procedure called NPX, which is an abbreviation for normalized protein expression, to minimize both intra-assay and inter-assay variation. Lastly, we then use the 5th library and replicate to look closely at system reproducibility. When comparing the replicate libraries, we found the correlation to be close to perfect, 0.999, here shown after NPX normalization in figure A on the left. In addition, by calculating the variation coefficient for each data point duplicated in the 2 runs, we found the average precision was very high, which you'll see in figure B on the right. In summary, the results clearly demonstrated high compatibility between the Olink Explore platform and the Element AVITI system. The data quality across the key metrics in side-by-side comparisons showed exceptionally high match count correlation, both pre- and post-normalization, and in replicate studies. I thank you for your time and welcome you to download the Olink Explore technical note that describes this work in greater detail from our website. Thank you.